So going forward here, Dr. Desai, I just wanted to just bring something up real quick. This past graduation, May of 2021, you received a gift, an honor in excellence in mentoring award. Could you explain that briefly?
Thanks, Drew, for bringing that. So, in addition to this, one of the joys of academics that I've had over the years is to serve as a mentor for PhD students. And I've been very, very, very fortunate to have had a number of excellent PhD students who are now working in industry and the regulatory agencies like the Food and Drug Administration here in the US and globally. So these are individuals who have gone on to make some really superb contributions, and I'm very proud of that. And some of them got together and nominated me for what's called the Excellence in Doctoral Mentoring Award at the university level, which I was fortunate to have received in this spring semester.
Thank you. It's an honor to have you, obtain that award, and I'm sure you're very proud of it, and your staff should be as well, knowing that they work for somebody who's working hard not only for the university, but for the students, and just the tight-knit that you guys have together as a core, as professors, it really does go a long way. So we're looking to continue that going forward. The next step I would like to do is kind of just ask the roundtable questions to each professor going forward. So to kick things off, Bill Seibel, I'd like to speak with you real quick.
Okay.
With you being in small molecule and drug discovery, could you explain that process and how it integrates with what you're doing teaching-wise?
A little bit unexpected, but I found this is sort of an introduction to the program and the class. So really, I guess we think about it as our goals are to help humanity, to find some sort of a cure of disease. And so the physician may look at this sort of a thing and see a mole, look at it. You can look at it and see some sort of aberrant function within the skin, and but that's not really close enough. I guess as a chemist, I like to think about it in molecular terms. And so if we start going into the cell, and even in the cell, look at what sorts of pathways are going on in that cell. Biologists like to write these things as sort of all these little globs looking at each other, but that's still not quite chemical enough. So actually all of these little components have structures to them. And really that's where you can start to design a drug. If you get into looking at the structure and then zoom in and really look at where this protein is actually serving its effect, down in here, you can actually look at that site and then actually use that structure to design something that's going to block that function of that protein and actually look at all the different interactions that might be going on here. So to me, that's really what the medicinal chemistry and the drug design of this thing is, and eventually you get a compound and you get a drug onto the market. The drug on the market is everybody else's responsibility. But if we want to look at the drug discovery process, I kind of think of it as clinical development. Development in general refers to human studies. I've never had anything to do with that, so I won't talk about that really at all. My life was really in the sort of the discovery side, which is everything before you get to humans. As Pankaj method talked about, the classic model for that is target identification and validation. Really, target identification is sorting through the disease state to really look at what proteins are aberrant in that disease. A lot of that is just sort of speculative, or maybe guilt by association, or even just circumstantial evidence. So you may have a variety of different protein targets that are suspected of being involved in the disease. The process of target validation is proving which of those actually modulates the disease. And you do that usually through making different genetic models or knockout studies, or a variety of different manipulations of the target to show that modulation of the target actually modulates the disease proportionately. Once you figure out what kind of protein you're trying to go after, usually you want to inhibit things because, like most things in life, it's easier to block something up than is to make it work better. We develop an assay. You have to have an assay to measure whether you're intervening with that target or not. So you go into this phase of assay development. And that's when you then move into hit identification. You can use the assay to screen compounds, and screening might be millions of compounds if you were a large pharma company and you don't really know what you're looking for. On the other hand, you have some good ideas on where to start. You can do that a little more efficiently. Through your screening program, if you find an active, it's great. And from there, go to hit the lead. One of the things that I hope students appreciate is a lot of the things that come out of assays are false positives, and so this hit the lead process is sort of separating the wheat from the chaff. And then once you have a compound that shows activity at your target and you know is actually a bona fide active, it goes into lead optimization, and that's the process usually dominated by medicinal chemistry and in vivo and in vitro biologists, wherein you make sequential modifications of a compound, hopefully being guided by the structure of the protein and some other tools to build in all the properties that a drug has. A drug has to be more than just active at the target. It has to be inactive at other targets, and it has to be available maybe in a pill form, so you have to be able to eat it. It has to be stable to your stomach, has to be absorbed, has to not be metabolized at least too fast. You don't want to never metabolize. It eventually has to be excreted. So you have to optimize all of those properties. And once one gets a balance between the potency and the selectivity and all of those other properties, you have a clinical candidate, and that goes into preclinical development. Preclinical development is generally sort of replicating some of the efficacy studies and then doing a lot of the toxicological studies under GLP conditions, good laboratory practices, which is really just rigorous protocols and documentation. And if that all looks good, you file with the FDA, a document called the Investigational New Drug Application, and you then can enter clinical trials. So that's really what I think of as the drug discovery process.
Thank you very much, Dr. Sci. Dr. Shelley, I understand that you have a you kind of coincide with this clinical aspect as well. Can you discuss on your end what they do once they enter your course?
Bill and I have been team teaching the course 80/20 drug discovery and biologic therapeutics now for quite a number of years. And Bill focuses primarily on the drug discovery aspect and on small molecules. I focus mostly on larger or biologic modalities that might be molecules like antibodies that you're typically used to hearing about, all those commercials for psoriatic arthritis with the antibodies you see on TV. But we also cover things like RNA interference, mRNA vaccines in general, viral vector gene therapies, cell therapies like CAR T that you may have also been hearing about. My session is a bit more of a survey of these kind of molecules, but I do get into some aspects of how they're discovered, developed, and then usually we have a segment of my portion of the course where we look at the unique aspects of regulatory, manufacturing, and clinical development of biologic therapeutics that differ perhaps from small molecule therapeutics. Like, for example, with vaccines, we specifically are looking for things like immunogenicity, which in most drugs, that's the last thing you want, is an immune response. But with a vaccine, that's exactly what you're looking for. Also with vaccines, we have to be concerned about something called scheduling, which is when do you actually give the vaccine? If it's a pediatric vaccine, where in the schedule of all the other vaccines that you give to children will your vaccine fit? So these are the kind of topics that I would typically be talking about in my portion of the course. And I guess one other comment I'd make, which is interesting, is that in biologics, academicians and industry tend to start from a slightly different perspective. The pipeline that Dr. Desai provided, in industry, if you were to go to a venture capitalist and say, I'm going to start with 10,000 compounds. I'm going to screen it down to one, and it's going to take me 12 years and $1 billion. That venture capitalist is going to run screaming for the hills, right? That's not really an appealing value proposition. But if I say, I'm going to start with a carefully crafted 1 or 2 lead compounds, I have an accelerated development pathway. I can find ways to streamline the cost of that clinical development and still do it safely. Now you're talking from an industry perspective. So I also try and bring that angle into it because that's where I come from.
Excellent. Thank you, Dr. Dan Shelley. So I'm now going to pass it to Richard Swen. So you had mentioned you'd been here prior to Dr. Desai's, before he came along. So, explain what you're doing in your course, and what is the goal that you want of your students when they have completed, your course at the end of the semester.
Yeah, thank you very much. Well, I'd like to refer to the sequence of key steps in the drug development program that Dr. Seibel recently just described in his brief intro. The aspect that is the focus of my part of Dr. Desai's course, which is preclinical phase 1 type of drug discovery. Is that correct, Dr. Desai?
Preclinical / non-clinical drug development. Yeah. So
yeah. Sorry. Always get those names mixed around a little bit. But a key part of the whole drug development sequence is triggered when a company has made a preliminary selection of the analogs or the compounds that are worthy of directing toward clinical trial. And of course, with that, a key part of that is there needs to be a number of studies conducted to confirm that the drug candidate is adequately safe as one interprets the results from in vitro and animal safety studies in order for it to be progressed into clinical trial. That usually entails a group of studies that there are 6 categories of which a compound must be then screened in order to generate those data to allow for a risk assessment for that proposed clinical trial. And very briefly, those categories are genotoxicity studies, safety pharmacology studies, single and repeat dose toxicology studies, reproductive and development toxicology studies, carcinogenicity studies, and then local toxicity studies. That's basically, you're looking at every organ system in the living creature that could be affected by the drug in a possibly a bad way. So what my part of the course is directed towards is educating people on those categories of studies and then specific examples within those categories so they understand the terminology, how to design a tox program, we'll call it the collection of studies a program, and then most importantly, in terms of making the risk assessment for a clinical trial, how to interpret the results in terms of effective dose or toxic dose from each of those studies in order to calculate the safe starting dose for a proposed clinical trial. So all this ties together. It's a very highly FDA regulated aspect of drug development. Meetings with FDA as a pre-IND type of meeting or elsewhere in the program always focus on the results from those studies, and the challenge to the company or the developer as to whether or not the data are worthy of going into a clinical trial and exposing these humans to this potential risk of toxicity. Of course, those clinical trials are leading toward an assessment of efficacy in whatever the disease state that the drug is targeted toward. So there's kind of an ongoing review of all of those data and I guess to simplify it, it's my segment of the program is really focusing on the toxicology program and the rationale for why certain studies are required.
Thank you, Dr. Schwen. So going forward, thanks for all the first phases and the clinical aspects for it. I kind of want to switch now to pharma economics, and that brings Dr. Jeff Guo into this conversation. Can you explain with borderline data and the information that is unknown, what makes your class important in drug development?
Thank you for your question. What we discuss in the class, related to the pharmacoeconomics, outcome research, or the phase 4 clinical study related to drug safety, it's really, in my opinion, it's more recent in the past 20, 30 years, because traditionally the pharmaceutical industry focus on the drug safety, efficacy, and quality. Up to the past 20, 30 years, more and more attention paid to economic consideration, whether or not the new drug developed can be accessible for the patient, can be reimbursed by the insurance, can be covered by the Medicaid, Medicare. So that consideration, the pharma have to conduct the pharmacoeconomic study to approve or show the evidence that the new drug is cost effective, it's cost beneficial, right? So that is what we discuss. But what we will share to our students a little bit more than that. We was talking about starting from a supply demand. What is the supply? That's our pharmaceutical industry. What is the demand? That's our patient, our medical doctor, pharmacist, insurance. So that's the demand. And what's the demand, supply, determine the price. And what is the problem about price, issue about the price, and policy related to our pharmaceutical industry. So that is what we discuss for the student. And I hope that answered your question.
Thank you very much. Anything else you want to add?
Oh yeah, I would say that after have this MSDD's training, we will have complete knowledge of from very beginning the pre-clinical drug discovery to clinical, all the way to post-marketing. So what I have been focused on is post-marketing, or when the products before the launch, from the research perspective, from policy perspective, from the safety perspective, monitoring, surveillance. So this kind of training, in my opinion, it's not traditionally in the pharmacy curriculum or medicine curriculum. It's especially in our MSDD program. We are the only program would coach our student to learn how drug be developed from very, very beginning all the way to deliver to the patient. So whole process. And this is a knowledge and this is education. We are very unique to provide that.
Thank you, Dr. Guo. So that's one of the things that we bring up when speaking with students, is because we'll get a lot of students that have been working in the drug field that either come from a Procter & Gamble or Johnson & Johnson or Pfizer, and they're at the 4th and 5th stage, so sales and manufacturing, and then they're sent back to the university to learn those first 3 phases of introduction, clinical trial, and then submitting to the FDA. That's what really makes this a very unique program. And you guys have done an excellent job so far relaying that information.
Thank you.
So continuing on, you know, the numbers side, I'm gonna now pass this over to Cindy Rothenberg. So with you being on the statistician side and teaching statistics, could you tell me how that plays a role in drug development?
Absolutely. So just as Dr. Guo talked about his course, which was on the pharmacoeconomics side and looking at the post-launch, you referred just a moment ago to like the phase 1, 2, 3 up through submission part of the program. And really my course is about really understanding the efficacy and safety of a compound as it goes through these individual phases. You know, as you think about the design of a given study and the particular potential measure that, you know, best kind of identifies the efficacy or safety of a compound, you know, each of those areas are going to kind of drive what are the right statistical procedures to analyze the data. So the course is really about what are the principles, the criteria for a good design. But then once you've engaged in the research, you've identified what that measure is and what that design is, well, how do you then, after you've collected the data, go about analyzing that? And so from an efficacy standpoint, you may have continuous data, such as, let's say, BMD in an osteoporosis type trial. You might have fracture. So you might have discrete data, or in a survival study, you'll have a time to event. So maybe time to progression or reoccurrence of some type of, you know, cancer or disease. Each of those requires a different type of analysis. So really the goal of my class is to enable students to understand how to design, how to analyze and interpret. And also one of the things that we do is we leverage statistical software, JUMP. So we leverage software to give them the tools to be able to do the analyses themselves. And that's really what's critical and the aim of my class. So that as students come out, they're going to be able to engage in their own research and then explore and analyze their own data and therefore interpret the efficacy and safety of that compound.
Excellent. Thank you, Dr. Rodenberg.
So, Joe,
when it comes, go ahead, Dr. Sci.
Quickly add, Dr. Rodenberg's class is one of our unique classes where it's clearly each session is half theoretical didactic, and the second half she really works with the students to give them hands-on exercises. So so that's one of the key things in statistical analysis. How do you actually start, you know, digging through and then working and you know, analyzing the data till you start doing it, you don't actually, you know, get the full concept. Mm-hmm. So that's one aspect of the curriculum that obviously I love. And the other aspect, that's something that Dr. Bapat does, is he actually, when the program was on site, he used to actually invite students to go to a real manufacturing facility at Procter & Gamble, or there's a device company that's a testing company, and he would invite the students to tour the testing facility. So as you will get to hear from Dr. Bapat, he will talk about how we are actually doing that, capturing that even virtually now with the online world.
Excellent. Thank you, Dr. Sci. Cindy, just a real quick question for you. In terms of math knowledge, what can students expect? Should they know calculus coming in? And, you know, give us a little insight on that.
So calculus is not required. That's a good question. It does not require calculus. What I try to do in the class is to, you know, explain the fundamentals, you know, so the fundamental theory and the understanding, but it is not requiring, say, integration, which obviously is a part of mathematical statistics. That's not a requirement. I like them to understand kind of the formulas. So I'm trying to think what would come before calculus. I mean, obviously algebra is a requirement. typical requirements. It's really, I try to explain the kind of give the underlying foundation and an understanding of it, but a more holistic understanding, so that as they look and they kind of understand first, how might these analyses be done by hand, so that they can then extend what they're learning in our class. But really then it's about understanding the software, understanding kind of the data that they have, and then how do I utilize the software to conduct the analyses. So it is not requiring complex mathematics to do that. One of the concepts that students probably do struggle a little bit with in like engineering and mathematics, that's a very deterministic. You have a mathematical formula, you put an input, you get a specific value. But in statistics, you have random variation. And so that's a big concept that I think students sometimes struggle with, but that's something we work around so that they can hopefully understand, again, like when I observe something, how likely is that? And therefore, or how unlikely is that? And therefore, how do I interpret whether or not now I've observed something unlikely enough that I definitely have a compound that is efficacious and safe. So I struggled a little bit with that answer, but hopefully that makes some sense.
No, thank you very much. Students are always wondering about math and how high they need to be in terms of their level of math, being a mathematician. So thank you very much. So next, I'm going to pass it to you, Dr. Brenna Carey. Trending in the industry is, what your goal is in your courses. Can you tell me what the trends are looking like while you're teaching your courses and so forth?
So I think the goals of my courses. So I teach the phase 2. So the second half of Dr. Desai's class on phase 1, phase 2 clinical research and design. And then the first part of the phase 3, phase 4 clinical research and design course. So I primarily focus on phase 2 and phase 3 courses. And so kind of what the objectives of my courses are to give the students kind of a comprehensive background of those critical trials that are necessary to show proof of concept, get the inner workings and the inner details of what the proper dose should be when they go into their pivotal trial, which is what's necessary for actual FDA approval of their drug. And I also have guest lecturers that come in that are specialists in their fields, so in epilepsy and infectious diseases, that kind of give them firsthand experience of developing a medication in certain fields, so that the students not only get facts about what are phase 2 and phase 3 clinical trials, but kind of have examples of certain trials in these different areas. And then I tried to, one of the projects that they do is to write a clinical protocol so they can then, use the concepts that they've learned in the phase 1, phase 2, and phase 3 courses to, choose a disease indication of their choice, and kind of develop an interventional protocol to, incorporate all the various aspects that have been covered in all of their courses, from the first year of drug development all the way up through, writing that pivotal clinical trial that's going to go to the FDA to get approval. And I try to incorporate, even the statistical plans and the information that they learn in Cindy's class, so that they have a complete, project at the end of the phase 3 session, when they then move into Dr. Guo's, post-marketing and pharmacoeconomics class.
Okay, thank you very much. Going next here, I'm going to pass it to Dr. Nitin Bapart. So, at the start of this conversation, Dr. Desai had mentioned that you're working with formulations and success stories. give us a success story from one of the students that's, you've taught, and then kind of just go into your course as well.
So another aspect of this course is we have the capstone project, and one student, after going through the manufacturing, he went up and took up two things. One was one student took up the continuous manufacturing, which is now a very new trend in pharmaceutical making. Generally, it has always been a batch process, so it's a new continuous way of making things. And then he took it up and he was talking to within his company how to bring about that process in part of inspiration, you can say, from learning it while he was taking our course. So those are some things what we do. And another things what we talk about on a daily basis is, especially through COVID, all of us are even suffering getting simple things, and we hear about supply chain. So while manufacturing of drugs is very regimented, what is also happening is that you sit back and look at it, the raw material sourcing. We talked about vaccines, and Dr. Shelley talked about it just now, and we are hearing about vaccine shortages. It's no different than what we talk about is these shortages, the manufacturing, how they are all coming together for even simple drugs. In the United States, we are facing drug shortages of, even before COVID, of other drugs which are generics and getting off and not being manufactured. So there are all these challenges, and the question is, oh, if that's a drug is already there, why can I not set up something and manufacture it? And so the challenges which come in just creating a GMP manufacturing facility are some things which we talk about, and we try to impress upon students. Even they could be in this field, or they could even be with a CRO evaluating how is the new drug coming along, and how are you making it, and importantly, how are you going to scale up and go to a different geography? Means apart from the studies which have to be done, will you even have a source of material to that? So these are just some aspects we try to allow the students to think about as to whichever role they are in, is how is the drug supply going to happen? How are they going to manufacture? And then there are the competition from generics, or for the biological things from the biosimilars. So those are big, these are big things which we talked about pharmacoeconomics, Dr. Guo talked about it. These are big things which impact where will you make the drug, and how will you make it, and what do you do about it. So those are some things which hopefully keeps it very interesting for the students.
Thank you, Dr. Papat. It's great information in terms of the last phases and what the students are going to see, because the capstone is always a question that they're coming through. They want to know what they're going to be learning, and that it kind of gives some insight on that course as well. And then finally, we have Dr. Ajayi Fumini. Thank you for your patience for being the last one, but being at the last phase of everything in the third phase, it's, it took me a while to get to you. But go ahead and explain your course and what and highlights that you bring to the table for the drug development program.
Well, thank you for that opportunity. My course is focused mainly on the clinical pharmacology aspect, and the main goal is to enable or equip the students to have a good understanding of how to obtain the relevant data that is necessary for the phase 3 clinical drug development program, where they will be developing the protocols in terms of what they need to have, the data that is necessary from phase 1 and phase 2, what they need to understand, the knowledge that they need to actually gain from even the preclinical work in terms of the pharmacogenomics, the pharmacogenetics, and how to bring all those together to have what we always call a rational drug development program for that particular candidate, whether it's a small molecule or large molecule, for us to have what I describe as the knowledge, because knowledge is power, for them to have a good understanding of what they need, which is must have, what they, nice to have, and the in-between, in such a way that they will have a cost and time effective program that is developed for or designed for that particular program. The other thing that we talk about is what is important to have for the marketing of that particular drug or candidate or biologics in terms of the data, because everything is data driven. All the claims in the package insert must be supported by data for the regulatory agencies to approve them. So they have to think along the way in terms of what data is needed and how to get those data in a cost and time effective manner. And that is the other thing that the students are usually very excited about, especially when we use case studies to look at what companies have done in the past, how they've been able to gather adequate and appropriate data to support the claims that they are making. And we also talk about when the data is not enough, not just in terms of the statistics aspect of it, but in terms of the planning process that may have gone wrong somewhere in terms of their thinking process. So knowledge is power is one of the things that we talk about, and the fact that we need to accumulate the knowledge right from the drug discovery throughout the drug development process in order to be able to make those wonderful plan that they need to make for that successful marketing of that particular product.
Thank you very much. That basically is, an introduction to everybody and your courses that you teach. It's very, very informative, and I do appreciate that. Dr. Desai, is there anything else that you want to speak right now?
Yeah, so if you don't mind, just give me 1 more minute to tie it up. So it's not really just development of new drugs and biologicals. Let me give you a couple of examples where our collective expertise really comes into play. For example, if a drug like, let's say, Lipitor, which is one of the most successful launches by Pfizer for lowering cholesterol level, that goes off patent. Well, one of the first things we'll do is we'll sit down with our intellectual property gurus who are teaching in the program to do what's called life cycle extension. We can think of making a new formulation, which is when we'll go to Dr. Bapat and say, okay, what can we do to make it, let's say, a sustained release formulation? Instead of taking it twice a day, can we do something where we can take it once a day or once a week? And of course, he's going to be talking to somebody like Dr. Ajay about what is the FDA's stance on that, and what kind of clinical pharmacology studies that we should really talk to Dr. Carey about in terms of, doing the phase 1 to phase, perhaps a phase 2 clinical trial if what we are talking about is requiring an efficacy assessment as well in some other cases. But then that's where Dr. Cindy Rottenberg's expertise in terms of can we statistically show that the new formulation is going to be actually able to do the same job as the compound, the innovator's compound that became generic. So there are a lot of issues pertaining to what we call generics, biosimilars, repurposing of drugs, then applications to therapeutic areas, whether they are orphan diseases. So there are a lot of nuances that are actually captured within the curriculum, which again, as I said, with the example of Lipitor, is a collective expertise that the industry obviously utilizes to launch newer products. And that's the expertise that we also have in this curriculum.